And in that event, you happen to lose your arms, legs, or perhaps even both. Pretty scary, right? Then imagine being introduced to a new and unheard of type of science that would allow you to reproduce those limbs naturally, almost as if they had never been lost in the first place. Would you do this? No questions asked. Today, I would like to inform you and share with you facts about stem cell research, the science behind it, the pros and cons, and also the ethical dilemma of this practice. Understanding the science behind stem cell research helps you to comprehend why it is such a controversial subject and why so much time and debate has gone into it. Stem cells are cells that have the ability to divide for infinite periods in culture and differentiate into specialized cells. There are two different types of stem cells: embryonic stem cells and adult stem cells. Also known as non-embryonic stem cells and somatic stem cells. Embryonic stem cells divide and differentiate into cells and tissues of the three primary germ layers, which are ectoderm, which gives rise to the nervous system, sensory organs, and skin. Mesoderm gives rise to the bone, muscle, and connective tissue and kidneys. And the endoderm gives rise to the respiratory structures and digestive organs. Embryonic stem cells can be used to divide and differentiate into pretty much any tissue in the human body and can divide indefinitely. They are taken out of the body from the blastocyst, which is an embryo that is a few days old, and because it is taken out of the body, the embryo is destroyed. They can also be obtained by extracting the cells from the morula, which is an embryo at an early stage of development that consists of cells in a solid ball, and the end the embryo is still destroyed. But morulas are easier for scientists to grow and manipulate, so the chance of creating a successful stem cell line is greater when a morula is used. But adult stem cells are a lot different than embryonic stem cells. When adult stem cells are extracted from the body, they can only maintain and repair the tissue in which they are located. Adult stem cells are found in many places in the body, including bone marrow, umbilical cord blood, muscle, and brain tissue. There are also genetically modified skin cells that can be morphed into a few dozen different type tissues throughout the body. They are called induced pluripotent, or just IPS. The capacity to divide is limited once the cells are extracted from the body. So embryonic stem cells are harvested during fetal development and exhibit pluripotency, which means they take the form of over 200 different types of tissue. This makes them ideal for regenerating tissues in humans because we cannot regenerate on our own certain tissues such as heart, spinal cord, and brain tissues. Here's a picture that shows how embryonic stem cells are taken out of the body and divided and differentiated into different organs and systems of the body. It is easy to recognize that stem cell research could possibly be a great thing and that the whole entire world would benefit from the good things that stem cell research would be involved with. The future of stem cell research is rooted around the term regeneration, which in this case refers to the ability to recreate lost or damaged tissues, organs, or limbs. Some animals have the ability to grow back lost arms, tails, and in some cases even their heads, but the current stem cells that we as humans are born with can only heal injuries, but it cannot replace missing pieces because our regeneration is limited. Regenerative medicine is a new field of medicine that is aimed to use stem cells to heal tissue in the body that cannot help itself. Some species Scientists hope to be able to replace organs and tissue that have been worn out by either age or injury. However, the new organ tissue will be genetically modified to last longer than the original component. But before scientists can perform these organ tissue transplants, the technique for isolating, growing, and expanding the cells must be perfected. Stem cells will also be used to study diseases by taking cells from individuals born with certain genetic disorders. Scientists will then be able to do further research with a reliable source of affected tissue and possibly come up with cures. This could lead to the curing of genetic disorders as Alzheimer's, muscular dystrophy, and cystic fibrosis, and also autoimmune diseases, multiple sclerosis and Crohn's disease. Stem cells can even help us learn more about cancer. Because like cancer cells, stem cells can grow very fast and remain undifferentiated. It's possible that some tumors are stem cells that have grown out of control. If we can learn that stem cells grow and differentiation works, it can help us better understand cancer and aid us in finding new medicine or even a cure for cancer. The possibilities for stem cell research is endless, and that is the beautiful thing about it. But not to get confused, there are definitely some things about stem cells research that are not so good. Along with the pros, there are also some cons. Just like any other practice, there are health risks that go along with stem cell use. Stem cells can turn cancerous if kept out of the body too long. They can produce telomerase, an enzyme that allows the cell to reproduce without any regulation. Adult stem cells that produce cancers are a driving force behind tumor growth, and even though they are 1 in 1 millionth of the entire cancer cell population. Stem cells live longer than regular cells, and they live just long enough to acquire a mutation, and many times a mutation is cancerous. A variety of cancers, leukemia, brain cancer, breast cancer, skin cancer, and colon cancer, can originate due to a mutation in adult stem cells. The assurance of if stem cells will work for everything they are said to work for is not fully known. Embryonic stem cells are the type of stem cells that scientists want and will go to extreme measures to get. There are no limitations to the possibilities that can come from embryonic stem cells. But with adult stem cells, there are limitations. They can only divide into cell tissue that is taken from and have a limit on the rate and quantity that it can multiply. While the embryonic stem cell is highly sought after, the ways of obtaining it are the reasons why stem cells have ethic dilemmas and why the situation tests people's morals. To obtain embryonic stem cells, you would have to take them out of the morula or the blastocyst, both of which are the embryo, which later turns into a human fetus. When the embryonic stem cells are taken out of the blastocyst or the morula, that automatically destroys the embryo, which, without being tampered with, would eventually turn into a human fetus, and within months would turn into a human baby. The point I'm trying to make is that by destroying the early stages of an embryo, you ultimately, in the end, kill a human being. The argument towards this is that when there is just an embryo that is either in its early stages or just a few days old, is it just considered an embryo with no life just floating around in the woman with stem cells inside of it, or is considered a human? Do we consider something that is soon to be a functioning human being, an actual person, even if it's not one yet? This is the moral dilemma. Is taking embryonic stem cells out of the embryo and in the end destroying it just the same as going down to the abortion clinic and destroying a baby? These are the questions and arguments that come up when embryonic stem cells are brought up. Even though we would benefit from them so much is it morally right is the big question. People would argue that if we are willing to destroy embryos for science and ignore the ethics and morality flaws behind that, then what other boundaries are we willing to cross? Thank you. And I would also like to thank my audience.
Monique Miller.
Melanie Hagan.
Janelle Hagan.